In this webinar, I will briefly present APC and VLE, then I will introduce our transduction and transfection process optimization workflow. Within this workflow, we combine the usage of three models. So I am going to give some additional information on these three models, being them CFD for mixing optimization and improvement on the recovery of viable cells. The second is on the transduction efficiency, so is where we are trying to improve the efficiency of the delivery of the genetic material to the targeted cells. And the third is a customized web application where we allow some additional scale-up to be done after the project has been completed through our platform iAchieve. At the end of the presentation, there will be a Q&A session as well. APC and VLE are sister companies based in Dublin, Ireland, and APC handles the research and development aspects of both pharmaceutical and biopharmaceutical processes, while VLE is a biologics GMP facility located in the same building. APC and VLE has enabled several companies across the globe to manufacture medicines in an accelerated fashion. In our small molecule labs, we have reactors varying from 25 mils to 5 liters, multiple PATs, including IR, Raman, PVM, FPIM. We also do continuous flow, isolation, and we also give support to formulation design. On the biologics function, there are multiple scale bioreactors, such as small shake flasks. We have a number with 24 bioreactors. We have multiple scales bioreactors up to 7 liters, and we also have a pilot plant with a 250 liters bioreactor. We also have a very competent analytical team with great capabilities. Just for a notion of size, only HPLCs we have more than 25. We have as well SEM, DLS, GCs, and all instruments required to guarantee the analysis of our samples. All this to deliver on our mission, which really is to accelerate the development of quality, life-changing medicines to patients. And we do it through applying cutting-edge technology and having a multidisciplinary, highly skilled, and very enthusiastic team. We have a multidisciplinary team, as I said. Many of us also holds a PhD, and we can support the life cycle of drug development from CMC and tech transfer to regulatory submissions. But today, I'm here to talk about our process and digital engineering. Within process and digital engineering, we support different projects such as analytical development, projects in small molecule and large molecule in general, safety, tech transfer, and others. We have diverse expertise within the group and an extensive software portfolio. All that to ensure that we are using the correct approach and the suitable tools for each challenge that is brought to us. Just to call out a few of these items, today I'm going to talk about CFD, DM and mixing models, but we do reaction kinetics modeling both by running the DOE experiments in the lab and also by fitting the models and running the simulations with the data collected. We do DOE, stats, and VDA analysis. Another big application in the small molecule would be solvent screening, crystal morphology prediction for crystallizations, and for that we use the atomistic and molecular simulations. We also have done a number of chemometric projects for different applications. We have modeled several unit operations such as filtration and centrifugation, and we also have supported various continuous improvement internally at APC and VLE and also externally for clients. Talking about our transfection and transduction model-driven optimization workflow, the first step is to design a digital twin of the actual system. To do that, we might request the blueprints of the equipment if they are available. In case of single-use reactors that can be sent to us, we can use them to take the measurements ourselves, or we can collaborate in a loop of requests until we get all the information we need to accurately represent the real system. In the sequence, we divide the volume occupied by the fluid into very small elements in a process that we call meshing. Navier-Stokes equations will be calculated within each element, so the larger the number of elements, the more detailed the simulation will be. We allow typically a maximum of 4 million elements to ensure that we're not using unnecessary computer power. Once we understand the geometry and the level of details of the simulations we should have, we include the process parameters in the simulation. Typically, they are the agitation rate, fluid density and viscosity, sparging rate, if applicable, working volume, and sometimes, in case of spraying systems, for example, we would request the pressure in the nozzle and the spraying time, for example. So the process conditions will really depend on the application and the system that we're using. Once we gather all the information on the current system, we can then run the fluid simulations. For example, here you'll see a shake flask on the left and a wave bag. Then we couple the fluid simulations with Discrete Element Method to simulate the particle movement in the system. Here, for example, you have a representation where we see the movement of the equipment, the particles, and the fluid. Once we understand the current process, we make a list of suggested optimizations that can be related to the geometry, such as change the impeller, change in the feed location, and so on, or change in the process conditions, such as the agitation rate, the sparging rate, and others. We take that list back to the client and agree upon what is feasible in the real system, and then once we agree about which simulations to run, we run those simulations. From the simulations, we obtain the optimal system with the optimal process parameters. This work is done in CFD and DEM, but in parallel, we add some mechanistic models to the simulations as well, such as the mixing time and Kolmogorov scale length, and those are very important for the recovery of viable cells. An additional model as well that we include is the workflow to create custom-made scale-up and scale-down tool, and that tool is really customized for each client, so it displays the reactors and impeller configurations possible to that processing specific and the scaling parameter that's most suitable for that process. I'll show an example of one of those apps at the end of the presentation. Well, this is the overview, and there is a lot in what I just mentioned. So let's talk a little bit in detail about CFD, DEM, and also the web app for scaleup. CFD stands for Computational Fluid Dynamics, and it is used to assess mixing performance and ultimately ensure recovery of viable cells. CFD is the most sophisticated approach for scale-up and for mixing optimizations through improvements of geometry and of the critical process parameters. Alternatives would be to scale up based on power per unit volume or power per unit mass, for example, but that doesn't take into account geometry differences across scales, nor the sparging effect, for example. Also, in case of bioreactors, the scale up or scale down by P over V might result in an agitation that causes an energy dissipation in the system that is high enough to damage the cells, but a simple P over V alone can't ensure recovery of viable cells. Because of that, CFD is a more complete analysis, really especially for scale-up and mixing assessment of biologic systems. So CFD is really the best option in order to obtain a visual representation of important parameters such as shear profiles, vortexing, velocity profiles. These profiles can also be extracted quantitatively, so you'd have the distribution. Through CFD, we can also access information that experimentation cannot provide. And by doing it, we can identify areas of concern, such as areas where fluid velocity is zero or close to it, which classify as a poorly mixed zone, so we should avoid those. We can also run several analyses from CFD outputs or couple CFD simulations with DEM, for example, and we can run mixing times, for example. We can use some of the parameters for the KLA studies, being KLA also heavily supported by laboratory experimentation as well. So I would like now to show how these visualizations and analysis, because there are so many, so how they can actually be used to ensure the recovery of viable cells in a transfection and transduction process. First of all, we need to ensure that the oxygen is being made available to the cells and that the media is being well circulated to ensure that the cells have access to the dissolved nutrients and oxygen in the system. Here we have on the left two graphics, one at 30 RPM and another at 50 RPM. Just by looking at these two images, I can tell that 50 RPM is giving lower recovery of viable cells. That is because these blue lines are the path lines of the oxygen bubbles. At 30 RPM, they are being well dispersed in the bulk volume. However, at 50 RPM, the agitation is stronger. The impeller seems to be downward pumping the fluid and the oxygen bubbles. The bubbles are then being pushed towards the bottom of the vessel and towards the side walls. And that means that this zone within those two regions here are zones where the oxygen is not being well distributed, and the cells within those regions might not survive. The velocity profile here on the right confirms the downward fluid flow. This type of plot is very useful to identify internal vortexes and dead zones where agitation is inefficient as well. Another important aspect of CFD simulations is the mixing time. For that, we use a tracer, which is a simulated die. Mixing is considered complete when we reach a COV below 1%. So we can visualize and quantify mixing times. We use this type of analysis to improve feeding strategies, for example. In this study, we analyzed a 5 liters bioreactor, and the mixing time was decreased by changing the overhead feeding to a bottom feeding. We also use this type of mixing analysis for comparison across mixing scales or to quantify mixing time optimization and mixing across process with different process conditions. Another standard item that we use to ensure recovery of viable cells through CFD is the Kolmogorov scale and the shear profiles. These are used to ensure that the energy and the momentum in the system are not high enough to damage the cells. The Kolmogorov scale is a measure of the energy dissipation in the system. The black horizontal line in the graphic on the left represents a cell diameter. The other three curves are the Kolmogorov scale lengths as we increase the agitation in these three scales. When the agitation is strong enough to cause the Kolmogorov scale length to be lower than the cell diameter, or we can see here by being below this black line, the cells will be damaged. So that's the region that we should avoid. And this analysis is done in combination with the shear profiles, qualitatively and quantitatively determined. Now that we have a couple of parameters to ensure recovery of viable cells, we need to think about the efficiency of the transduction or transfection processes. How can we ensure that the genetic material is actually being delivered to the targeted cells? For that, we use DEM. DEM stands for Discrete Element Method, and this is a mesh-free method that solves Newton's equations of motion for each particle. What we require to run a DEM simulation is the equipment geometry and the fluid dynamic profile from the CFD simulations. But during the CFD simulations, we only include some process parameters, fluid properties, and system geometry. So now we need to inform the simulations about the particles. And for that, we need to inform the particle size, shape, and density. We also need to understand how many of these particles we have in the system, and in some cases, what is the zeta potential when electrostatic energy plays an important role. When I say particles here, I'm referring to cells and the genetic material, be it a virus or a lipid complex, for example. If you don't know what is their size, shape, or density, our analytical team is able to perform analysis and determine these values. For some systems as well, we might also find some values, some reference values in the literature. On the left, we have an example of the power of the simulations. We can see the movement of the equipment, the movement of the fluid, and the movement of the particles. Also, the particles have different shapes and size. On the right, we can see an example of a project we did recently where we were transducting cells with viruses. We can see that the viruses had considerably smaller diameters in relation to the cells. A note here is that we can model particles even if they have different shapes. They don't need to be always spheres. We can also use a particle size distribution instead of single values. Here are some additional visualizations about the particles and the collisions. On the left, we can see a deposition of particles in the bottom of the system. The density is visibly greater in the bottom in comparison to the top, where we can see some spaces between the particles. These might be due to poor mixing, and that's affecting this deposition of particles in the bottom. These can also flag other improvement opportunities in the system. So it's always a good sign and a good feature to be able to see where the particles are being deposited or where they're traveling to. Here on the right, in green, we see only the points of collisions between cells and viruses. The message here is that we can produce videos, images, and quantify exactly how many collisions are happening at each time point and where. This is extremely powerful when we want to ensure that the genetic material is actually being delivered to the targeted cells. In this graphic here, for example, the system C is the one that resulted in the greater number of collisions between cells and viruses. That might be, for example, using a different impeller in comparison to the system A, and by quantifying the collisions between cells and viruses, we see that that's actually the optimal system to be running. So far, we've talked about how CFD can ensure good mixing and recovery of viable cells, and how DM can be used to ensure transfection and transduction efficiency. Therefore, if we combine both of them, we can ensure recovery of viable transduced cells. Now, I would like to talk about custom-made web applications that we develop at APC and host in iAchieve. iAchieve is our knowledge management platform where we save all the work we do. Our lab notebooks are actually electronic notebooks, and all experiments we run in the lab or that we simulate go to iAchieve daily. For the client, it is as if you're sitting beside us and you have real-time access to everything we're doing or planning to do. And all experiments are contextualized. That means that each experiment is linked to one workbench item, and that item has to be linked to a project objective. So everything that we do has a reason to be done. I won't go in much details about iAchieve today. I just want to mention that through iAchieve, we provide all the data, insights, discussions, and conclusions of all our work. And we also provide a report at the end of the project. Besides all that, we provide the web app through iAchieve, so you can also go through some of the models yourself. This is an example of a mixing web application. We have two columns here. The left is the control vessel. On the right, we have the vessel that we want to do the scaling to and find out the agitation to be used in this scaled up or scaled down vessel. Each project would have a customizable version of this, which includes the reactor name, the impeller type, clearance, bulk, fluid density and viscosity, field volume, and so on. Some parameters about the cells as well, because they're used for the Kolmogorov calculations. And those parameters are, for example, cell diameter and density. Having this information, you can choose to scale up based on impeller average shear, P over V, micromixing time, and Kolmogorov scale length. Don't worry if you add incorrect values. It will give warnings because it recognizes funny inputs. For example, if you choose a 7-liter bioreactor and type in the working volume of 8 liters, an error message will pop up to draw your attention to that value. So this is how to set up the scale up, and these are the results obtained. We can see that all the scale parameters were calculated, and one of the parameters was kept constant because that was the parameter we chose to do the scale up. This tool is particularly useful if you might be operating at slightly different working volumes in the future and need to do a quick check on the recommended agitation based on the designed scaled up strategy. In conclusion, this work combined 3 levels of modeling: CFD, DEM, and the production of a web application for scale-up. The CFD part of the workflow has been applied to more than 150 different bioreactors, including bioreactors or biological systems that had an unusual geometry because they're systems in development. The DEM part of the workflow has been successfully used in a transduction process, and while we were running that transduction project, we increased the recovery of viable transfected cells in 200%. We did that by optimizing the CPPs to maximize the number of collisions between cells and virus to ensure the delivery of the genetic material to these target cells, and we did that while keeping all the suitable conditions for the survival of these cells. Then finally, we did the scaling tool in form of a web application hosted in iAchieve, and that was used in multiple commercial projects for scaling up and has received very good feedback from scientists and engineers. Thanks everybody for listening. Now I'll be very happy to answer a few questions.